Hello everyone, I'm Dr. M. Venkateswarulu. Here I'll talk about my research on enzyme-responsive nanoscale capsules for anti-cancer drug delivery. These are tiny smart carriers that release drugs in response to enzymes, aiming for more targeted cancer treatment. This project was carried out under the guidance of Professor Artur R. Stefankovic and is funded by the POLONEZ BIS program. Here we show the challenges in chemotherapy. As we know, cancer remains one of the leading causes of death worldwide. The World Health Organization projecting up to 22 million new cases per year by 2030. The social and economic costs of the disease are profound. In 2012, the first estimate of the economic burden of cancer in Europe was publicized at the European Society for Medical Oncology ESMO Congress in Vienna, with the total cost exceeding 124 billion euros per year. Chemotherapy has been known for decades. Anti-cancer drugs are used worldwide for cancer treatment. However, they have many drawbacks, including side effects like toxic to normal cells, poor solubility in water, poor stability in physiological environment, and dose limiting. These drawbacks limit effectiveness and cause side effects. Moreover, these indicates that the selective inhibition of cancer remains a challenge in chemotherapy. To overcome these issues, stimuli-responsive nanocarriers including liposomes, micelles, MOFs, metal organic cages and dendrimers have been developed that respond to triggers like pH, temperature, light, and redox. However, the clinical use of these nanocarriers is still limited by drug resistance. Moreover, stimuli-responsive supramolecular nanocarriers received few drawbacks, including non-targeting, uncontrolled drug release, limited solubility and stability in physiological environment. These drawbacks limit effectiveness and cause side effects. To overcome these issues, enzyme-responsive nanocarriers have been developed. Enzyme-responsive systems are especially promising because enzymes are naturally overexpressed in cancer cells. My project aims to design and develop new enzyme-responsive nanomaterials for controlled drug delivery. We propose to develop a series of supramolecular metal organic capsules, which feature enzyme-cleavable groups including disulfide bridged, carboxylate esters, phosphate ester, or azo-bridged, which will be cleaved by overexpressed enzymes in cancerous cells and release an encapsulated anti-cancer drug and decrease the detrimental side effects associated with anti-cancer drug-based cancer treatment. Why we choose metal organic cages? Metal organic cages are ideal for this purpose because they're easy synthesis, soluble in water, have three-dimensional hydrophobic cavities to bind guest and modulate the properties like solubility and stability, etc. This makes them versatile carriers that can be engineered to respond to enzymes. Our plan is to design enzyme-responsive cages built from specific ligands in metal centers. We have selected two classes of MOC. First one is a platinum-2-based MOCs. In this class, platinum-2 metal centers and polar nitrate NO3 counter-ions produce water-soluble assemblies and ensuring good bioavailability. And also, platinum-2 complexes are also known as chemotherapeutics. Second is another well-known class of cobalt-3 MOCs. Cobalt-3 complexes are kinetically inert and more stable in biological media. The use of cobalt-3 is advantageous as it allows the formation of robust cages by exploiting an assembly followed by locking method. This approach utilizes the dynamics of cobalt-2 to facilitate cage formation under standard thermodynamic control, and then the MOC is locked by oxidizing the metal ions to inert cobalt-3. We will introduce the same cleavable functional groups into the cobalt MOC to generate enzyme-responsive properties. Following confirmation that the host has been synthesized, we will move to study the encapsulation of anti-cancer drug molecules and enzyme-responsive drug release in solution, which will be supported by appropriately chosen analytical techniques like NMR spectroscopy, UV vis, and fluorescence spectrometry. Following confirmation that an enzyme-responsive host-guest construct has been synthesized, we will move to cell studies using mammalian cancer cell lines and leading to cancer cell death. Here is the innovative aspects of our project. This is the first time novel enzyme-responsive metal organic cages are being developed for chemotherapy. They address the disadvantages of conventional chemotherapy, open possibilities for selective drug delivery in other diseases, and create a new bridge between supramolecular chemistry and medicinal bio-inorganic chemistry. I would like to thank Professor Artur R. Stefankovic and the group members for their insightful discussions and productive team meetings, as well as the Center for Advanced Technologies at Adam Mickiewicz University in Poznan for providing the instrumental facilities. I would also like to express my sincere gratitude to Professor Cheng Zhang for their invaluable collaboration and support. I would like to express my gratitude to the Polish National Science Center (NCN) and the European Commission for co-funding the POLONEZ BIS Fellowship Program under the Marie Skłodowska-Curie Co-Fund Grant. Thank you.